This video is for The Living Earth, Unit 4.4, Genetic Disorders and Technology. Today's essential question is, what are the uses, benefits, and risks of genetic testing and gene therapy? Today, we're diving into a fascinating and complex topic, genetic testing and gene therapy. These are powerful tools in modern biology, but they come with important considerations. Remember, our essential question today is, what are the uses, benefits, and risks of these technologies? By the end of this video, you should be able to explain how genetic testing identifies disease-causing alleles, how gene therapy aims to treat genetic disorders, and what the Human Genome Project has taught us about our DNA. Genetic testing and gene therapy, they represent significant advancements in medical science. Genetic testing allows us to detect specific alleles that may lead to genetic disorders, enabling early intervention and informed decision-making. Gene therapy, on the other hand, offers the potential to correct these genetic anomalies by introducing healthy genes into a patient's cells. While these technologies hold immense promise, they also present challenges and limitations that we must consider. So let's explore the various methods of genetic testing. First one, enzyme analysis. This method evaluates the activity levels of specific enzymes. For instance, in Tay-Sachs disease, a deficiency in a particular enzyme can be detected through enzyme analysis, indicating a potential carrier status. Next one, mutation analysis. This technique involves examining DNA for specific mutations known to cause diseases. For example, mutations in the CFTR gene are associated with cystic fibrosis. Chromosome examination, also known as karyotyping. This method involves visualizing chromosomes under a microscope to detect large-scale abnormalities, such as an extra chromosome 21 found in individuals with Down syndrome. And lastly, prenatal testing. Procedures like amniocentesis and chorionic villus sampling allow the detection of genetic disorders in a fetus by analyzing samples of the amniotic fluid or placental tissue.
Genetic testing involves taking a sample of a person's DNA and collecting information about all the loci throughout the genome. Some of those genes might predispose to certain cancers. Other genes may influence whether or not you'll gain weight in a certain way, or have hypertension, or cholesterol problems. So that's information that might be useful. Do you want that information? And that's some of the risks. Can you handle it knowing that you might get cancer someday, or do you want to be ready?
Gene therapy is a groundbreaking approach that aims to treat genetic disorders by correcting the underlying genetic defects. This process involves replacing diseased genes, which by introducing healthy copies of the genes to compensate for malfunctioning ones, we can replace the diseased genes. Then using virus vectors, harmless viruses are engineered to carry the healthy gene into the patient's cells. Then injecting healthy cells. These modified cells are introduced into the patient's tissues, where they can integrate and function. And lastly, multiplying and producing proteins. The new cells replicate and produce the necessary proteins that were previously deficient, alleviating the symptoms of the disorder.
So gene therapy conceptually is very simple. It should work 100% of the time, and when you can get it delivered, it does. And the problem is the delivery to enough cells. So what needs to get better is the vehicle, right? And that's what many people are working on, is how do we make a better Ferrari, right? Can we make it faster? Can we make it stronger? Can we make it more penetrant? If I need to modify your blood cells, and I can take some of your stem cells, the cells that make all your blood, and get it in there and re-deliver it to you, I'm probably going to have a better chance at impacting than if I have to get it into everything in your liver, right? Because I can't really mesh up your liver and deliver it and put your liver back. So then now your vehicle has to be able to get through and get into all the right cells. So the promise is huge. Right now the problems are delivery. It's not a perfect system, but it's pretty good and it's getting better. But delivery remains our problem.
The Human Genome Project was an ambitious international effort to map all the genes in the human genome. Initiated in 1990, the project aimed to identify and sequence the approximately 20,500 human genes. By the year 2000, a rough draft was completed, and by 2003, the entire human genome was successfully sequenced. This monumental achievement has paved the way for numerous advancements in medicine and biology. The completion of the Human Genome Project has led to several significant benefits. Of them, better medicines. Understanding the genetic basis of diseases has facilitated the development of targeted therapies and personalized medicine. Next, disease understanding. Identifying genes associated with diseases like cancer has improved diagnostic and treatment strategies. Next, forensic investigation. DNA profiling has become a crucial tool in criminal investigations and legal proceedings. Next up, agricultural advances. Sequencing the genomes of crops has enabled the development of genetically modified plants with improved yield and resistance to pests. While genetic testing and gene therapy offer numerous benefits, they also represent challenges. First, accuracy. Genetic tests may yield false positives or negatives, leading to potential misdiagnoses. Next up, gene insertion errors. Introducing new genes carries the risk of disrupting other essential genes, potentially causing unintended consequences. And lastly, genetic counseling. Given the complexity of genetic information, counseling is essential to help individuals understand their risks and make informed decisions. At this point, you should be able to generate a full and appropriate response to today's essential question. What are the uses, benefits, and risks of genetic testing and gene therapy?